Okay, folks, so this week we're going to focus on the immune response against pathogens. So that's the immune response against bacteria, viruses, parasites, and we'll also focus on the type of immune response that takes place when you get a disturbance at the mucosal surface. So this week will be broken up into two presentations. The first presentation, which I'll deliver now, will give a general holistic overview of the Th1, 2, and 17 responses. And we'll also delve a little into B cell activation, T cell activation, and then the concept of vaccinology. The second presentation, which will also be uploaded in Blackboard Learn, will go into a lot more detail in the Th1, 2, and 17 responses. So this presentation that I'm about to deliver is to help you try and get the holistic overview, so it'll make the next presentation slightly easier because you've been exposed to some of the content already. So in terms of what I would expect you to know by the end of this, I would expect you to be able to discuss the generation and function of Th1, 2, and 17 cells and the associated immune responses. I would hope that you'll be able to describe the interactions between CD4 helper T cells and B cells that would subsequently lead to the production of a high affinity antibody. I would also hope that you'll be able to describe the generation of a Th1 response and the effector function of the associated CD8 T cells. I would hope you'll be able to describe the immune response associated with mucosal disturbances. And then also I would hope that you'll be able to describe the immune responses involved in the control of all pathogenic infections. And lastly, for this presentation, I hope you'd have some understanding of the approaches to vaccinology. Okay, folks, so what I'm going to start off by doing is giving a quick synoptic overview of B cell development and then T cell development. So in terms of B cell development, so B cells are produced and mature in the bone marrow. And in the bone marrow, they rearrange their B cell receptor genes. And once they've rearranged their B cell receptor genes, those cells will be tested to ensure that the B cell receptor works functionally. And that's done through a process of positive selection. But it'll also be checked to ensure that the B cells don't react against self-antigen. And that's done through a process called negative selection. And at the end of that process, what happens is the immature B cell leaves the bone marrow, producing IgM, and it makes its way towards the spleen. And in the spleen, what will happen is it'll go through another round of checks to ensure that it doesn't respond against self-antigen. And at the end of that process, it leaves the spleen as a naive, mature B cell. Now, what that means is the B cell's mature and is ready to engage with antigen, but it hasn't yet been activated by antigen. So this follicular B cell will leave the spleen and make its way towards the lymph node. And it'll go into the lymph node and reside within the B cell area, the cortex. And in this area, it awaits for antigen to arrive in the lymph node. And when it arrives, the B cell will become activated through a series of processes which we'll cover later in the lecture. But once it becomes activated, it then enters the follicle within the cortex of the lymph node. And when it enters the follicle, it starts to undergo a process of somatic hypermutation, where it undergoes another round of mutation of its B cell receptor genes, with the outlook that it can hopefully develop a B cell receptor immunoglobulin that has an even better, stronger affinity for the antigen that it wants to target. And at the end of that process, it will then class switch to the antibody of choice, i.e. MAD or MADGRE. And at the end of that process, it can either become a memory cell, which means that if we encounter this pathogen again, these memory cells will kick into action rapidly, enabling us to have a very rapid adaptive response a second after the first infection. Or it can become, more importantly, a plasma cell. And the plasma cell is a B cell that will produce massive amounts of antibody. That antibody will then attach to the pathogen and enable its subsequent elimination. So that's a general holistic overview of B cell maturation. Now, In terms of T cells, pre-T cells are produced in the bone marrow, but these pre-T cells leave the bone marrow and make their way to the thymus. And in the thymus, that's where it undergoes positive and negative selection. Okay, so these pre-T cells will mature and develop, and it exit the thymus as either a CD4 naive CD4 T cell or a naive CD8 T cell. Okay, and these cells will then make their way to the paracortex in the lymph node. And in the lymph node, it'll wait for antigen to be presented to it. And the cell that's going to do that in terms of presenting the antigen is a dendritic cell, as indicated here. And what will happen is a dendritic cell will activate a CD4 cell and a CD8 cell. The CD4 cell will then provide help to the CD8 cell, and subsequently the CD8 cell will go to the site of infection and eliminate the infected cells. Okay, so that's a general holistic overview. Okay, and this is just the written summary of that process. Okay, and we're going to focus now as Th1 to 17, and we're also going to cover immune evasion as well. Okay, so in terms of the adaptive responses to different pathogens, if we have a viral infection, we need to recruit we need to initially recruit a CD4 helper T cell. Okay, so no matter what type of infection we have, the first thing that needs to happen is we need to recruit a helper cell for that response. So what will happen is the dendritic cell will recruit a helper cell, and in this case, it will result in the stimulation of a Th1 helper cell. Okay, and that Th1 helper cell will then provide help to the other effector cells, and in this case, the effector cell for a viral infection is a CD8 T cell. So a CD8 T cell will be recruited as an effector cell, as will a macrophage. Okay, so what I need you to appreciate is when we get a viral infection, the cells that play an important role are CD4 helper T cells, which are Th1 cells, and the effector cells are CD8 T cells and macrophages. Okay, now if we have an extracellular bacterial infection, or if we have an extracellular pathogen present, then what we require is a helper T cell, of course, again, and in this case it will be a Th2 helper T cell. Okay, and the effector cells that this Th2 helper T cell will provide help to are B cells and phagocytes. Okay, so the effector cells for dealing with extracellular bacteria are B cells, which will produce antibody. The antibody will bind to the pathogen, and then the phagocytes will start to phagocytose those antibody antigen complexes. Okay, so they're the cells that are required for extracellular bacterial infections. In terms of parasitic infections, they too require a Th2 response, but it's a bit more specific in that in a parasitic infection, it requires the Th2 CD4 helper T cell to initiate IgE-mediated response. And in terms of effector cells, it requires IgE producing B cells, and the other cells that it requires are mast cells, eosinophils, and basophils. Okay, so there's a bit more going on with the parasitic infection, and again we'll cover this in a wee bit later on in the lecture. In terms of what will happen if we get a disturbance at the mucosal barrier, because of course the mucosal barrier is the gateway to our systemic systemic tissue, so it's important that we maintain the mucosal barrier. But if it does become defective, then the helper cell that we need to stimulate is a Th17 helper cell. Okay, and what the Th17 helper cell will do will stimulate a vast amount of cytokines and chemokines, which will result in an influx of neutrophils and monocytes and other immune cells to try and keep the massive amount of pathogens entering the mucosal barrier at bay, and subsequently will result in the repair of the mucosal barrier. Okay, so that's a sort of very basic look at how we deal with each individual pathogen. So we'll go into a little bit more detail in terms of a viral response, a Th1 response. Okay, so here we have cells, could be in the liver, kidney, wherever it is, but we have cells that have been infected with a virus. Okay, here we go. And the first thing those infected cells will do will be to present that viral antigen on MHC1. Okay, so the cell that's actually infected will present the viral particle on MHC1, and that's important for the last stage of this process. Okay, but what then will happen is one of these cells will die and subsequently release viral particles outside into the extracellular space. And what then happens is that the dendritic cell will come along and it will take up this virus, which is now an extracellular pathogen because it's existing outside the cell. It'll take it up into its cell, break it down, and present the antigen on MHC2. Okay, and that dendritic cell presenting the viral antigen on MHC2 will make its way to the lymph node, and when it reaches the lymph node, it will present the viral antigen to a CD4 T helper cell. Okay, and it will stimulate the CD4 helper T cell, and that CD4 helper T cell will then become a Th1 cell. Okay, and at the same time, the dendritic cell will also present the same viral antigen on MHC1 to enable it to activate a CD8 cell. Okay, so what we have is we have a activation of a Th1 CD4 helper T cell, and now we have activation of a CD8 cell, and then the CD4 Th1 CD4 cell then produces a range of growth factors which will help further activate a CD8 and provide it, make it, equip it with the tools that will enable it to deal with the viral infection. So the CD8 cell then leaves the lymph node and makes its way back to the site of infection, where the infected cells are expressing the viral antigen on MHC1, and it then will use its T cell receptor to engage with a virally infected cell, and subsequently release perforin and granzyme, which will enable the infected cells to lyse and be degraded. And that is essentially how the Th1 response goes in terms of elimination of virally infected cells. Okay, folks? So, now, Th2 response in terms of a bacterial infection. So, here we have a normal healthy tissue, and for one reason or another, it becomes infected with bacteria, as you can see here. And what will happen is a dendritic cell within that tissue will come along, take up the bacteria because it's an extracellular pathogen, break it down and make its way to the lymph node. And at the same time, you'll also have, whilst you'll have the dendritic cell bringing the antigen to the lymph node, you'll also have some of these bacterial components making their way through to the lymph node as well. And when we arrive at the lymph node, the dendritic cell will present the viral antigen to a CD4 T helper cell. Okay, CD4 T helper cell will recognize the pathogen using its T cell receptor and subsequently become a Th2 cell. Okay. Separately, what will happen, that bacteria that managed to make its way to the lymph node will come along and attach to the B cell receptor of the B cell. And that will start the process of B cell activation. But the B cell will need further activation. And what will happen is it will take the bacteria will attach to the B cell receptor, and the B cell will internalize the B cell receptor with the attached bacteria, break down the bacteria, and present it on MHC2. Okay? And what the B cell will then do will be to present that antigen to the Th2 cell. And at that stage, what will happen is this interaction will subsequently lead to the production of growth factors by the Th2 cell to provide help to the B cell. The B cell will then make its way towards the follicle, and in the follicle, the B cell will start to undergo the process of somatic hypermutation, and with the hope that it'll produce a B cell receptor or immunoglobulin with a much higher affinity for the antigen in question, and it will then undergo class switching, as we've discussed before, and will then leave the follicle and produce massive amounts of antibody as a plasma cell. And then the antibody will arrive at the site of infection and bind to the individual bacteria, essentially opsonizing it or marking them for destruction. Then you'll get the influx of neutrophils, and neutrophils will use these little Fc receptors that you can see here. And these Fc receptors will enable the neutrophil to attach to the Fc region of the antibody and subsequently eliminate the pathogen by phagocytosing it. Okay? At the end of that process, then the tissue will then go back to being healthy. Okay? Now, it's very similar in terms of a parasitic infection. The exact same process will happen, and the dendritic cell will pick up part of the parasitic antigen. It will stimulate the CD4 T helper cell, become a Th2 cell, and then the parasitic antigen will also stimulate the B cell, and the B cell in this case, once it's stimulated by once the B cell is stimulated by the parasitic antigen and has undergone somatic hypermutation, and then we get a plasma cell, what will happen in the follicle is it will class switch the IgE. We need IgE antibodies for a parasitic infection. In this case, we have a worm. Okay, and the IgE will come along and opsonize or attach to the parasite. And then what we'll have is eosinophils, mast cells, and basophils all coming along and using their FCE receptor. And using their FCE receptor, it will attach to the FC portion of the IgE. And after that, what will happen is each one of those cells will release their lytic enzymes outside the cell, and it will destroy the parasite. Okay, so that's a parasitic infection. And then at the end, the parasite's gone, and we then have healthy tissue. Now, in terms of a mucosal infection, we require a Th17 response. So this is a general setup, and in normal healthy tissue at the mucosal surface, you'll have the epithelial surface here, which basically keeps all these environmental bugs on the outside and helps keeps everything well balanced. And here you have the blood where you have neutrophils, but of course, because there's no infection, there's no need to have neutrophils in the tissue. So what can happen is the mucosal barrier can become defective, and that essentially then just creates a gateway for mass amounts of microbes that are present in the environment to start making their way into the tissue. And that creates a real problem. So what will happen is the dendritic cell will collect some of this antigen, break down the antigen, and subsequently present it to in the lymph node or even more locally. In this case, it'll present the antigen to a CD4 helper T cell, And that CD4 helper T cell will then subsequently become a Th17 cell. And what will happen then is the Th17 cell will start to produce a whole range of different chemokines and cytokines that will stimulate the processes of inflammation. And that will subsequently lead to increased vasodilation, as you can see here, but also increased chemotaxis, which will subsequently lead mass amounts of neutrophils to enter the tissue and subsequently phagocytose the pathogen. And the pathogen then slowly disappears. And so then once the mucosal barrier is repaired, then the neutrophil disappears and the tissue then becomes healthy again. So that's a Th17 response, where basically we want to avoid this mass influx of pathogens. So the Th17 cell will induce a very accelerated influx of neutrophils to help deal with that process and maintain that mucosal barrier. Okay, so folks, that's the basic concept of each of those immune responses. What I'm going to cover now is the specifics of CD4 T cell activation and the specifics of CD8 cell activation and the specifics of B cell activation. So we'll start with CD4 T cell activation. Okay, so in this case, it will be the exogenous pathway. Okay, so the exogenous antigen, whether it's a bacteria or a virus that's been released from a dead cell, is taken up into the cell in an endosome. It then becomes acidified so that the antigen can start to be degraded, and then it starts to it then awaits for the MHC to arrive. And what happens is in the endoplasmic reticulum, the MHC is associated with the invariant chain. And the reason for that it's associated with the invariant chain is to avoid any endogenous antigen binding to the MHC2, which we don't want. We want to save that MHC2 for the exogenous antigen. So when the MHC then leaves the endoplasmic reticulum with the invariant chain attached, makes its way through the Golgi, and then when it interacts with the endosome, it then starts to get cleaved by cathepsin S. And what cathepsin S will do is start to prepare the removal of invariant chain. So it'll remove a segment of invariant chain, but it'll leave a component called CLIP, which will ensure that the MHC2 antigen binding region is still covered. And then when it progresses to the MHC2 loading compartment, then HLA-DM will facilitate the removal of CLIP and the addition of the epitope or antigen. And that epitope or antigen will then make its way to the surface and be presented to a CD4 helper T cell. Now, what happens when the antigen presenting cell presents the antigen? How does that then lead to activation of the T cell? Well, the first thing that happens is the MHC2 presents the antigen to the T cell receptor, and the T cell receptor is designed to bind to this specific antigen, and that provides signal 1. Okay, but signal 1 isn't enough to stimulate the activation of this CD4 helper T cell. What then will happen is the T cell will express the ligand CD28, and the dendritic cell will express the ligand B7. And when them two ligands interact, you get signal 2. And essentially, signal 1 and signal 2 together will subsequently activate the CD4 helper T cell. Okay, folks, so that's how T cell activation takes place. Signal 1 and signal 2. Okay, CD8 T cells, very much the same thing. The only difference is that the antigen presentation takes place through the MHC1 endogenous antigen presenting pathway. I'm not going to go through all the details for now, but it is present in last week's notes. So basically, the viral antigen gets broken down attached to MHC1 and presented on the surface. And when the dendritic cell presents it on the surface, then the CD8 T cell uses its T cell receptor to bind to this antigen that it's able to recognize. And at that point, that gives us signal 1 for CD8 cell. And same again, what happens then is the T cell will express the ligand CD28, dendritic cell will express B7, and that interaction will give us signal 2. And that will subsequently, that these two signals together will provide activation of the CD8 cytotoxic T cell. Okay, now B cell activation. It's slightly different. Okay, in this case, we've used salmonella, B cell that responds against salmonella as an example. Okay, so here we have an inactivated B cell that's waiting for antigen to arrive. Okay, and what happens is the antigen reaches the lymph node, binds to the B cell receptor. The B cell, at that point, the binding of the antigen to the B cell receptor gives that B cell signal 1. Okay, so B cell activation has started, but it's not complete yet. So what then happens is the B cell will internalize the antigen, break it down, and present it on MHC2. It will then start to go through the process of interacting with a Th2 CD4 helper T cell. So that CD4 helper T cell will recognize the antigen that we're hoping to have an immune response against using its T cell receptor. And then it will then, at that point, provide signal 2 to the B cell by expressing CD4 ligand, which binds the CD40 ligand, which will interact with CD40, which is expressed by the B cell. And that is what gives the B cell the second signal for activation. CD40 associated with CD40 ligand. Okay? And that is basically how you activate a B cell. Now, folks, that is a very basic overview. And I hope it gives you the very fundamental concept before you go to the second recording, because the second recording goes into a lot more detail. So hopefully this very holistic overview of those processes might make it slightly easier to deal with the complexity of the second recording. What I'm going to deal with now is the diversity of viruses, and just a very short introduction. You know, the viruses are extremely diverse in their ability to infect, persist, and initiate disease in host. In terms of entry, mucosal surfaces are punctured skin, such as insect bites and needles, basically gives the avenue for the virus to infect the bloodstream. So that's one way we get direct entry. And then in terms of spread, then you can have the replication, which usually occurs at the epithelial surfaces, and then subsequently manages to spread and make its way to the blood. And of course, once it reaches the blood, it has the capacity to reach all the systemic tissues. And there's two sort of types of viral infection that you may or may not be aware of. One is a sort of acute infection where you get infected, you recover, and the virus gets eliminated. But the more insidious aspect of viral infections is their ability to persist, and that concept is called latency, where some viruses are able to persist in a latent form after the acute infection is resolved and can reactivate to produce new infectious variants later on. And this is a sort of concept that you'd probably be associated with the herpes virus for things like cold sores, where basically you get infected with the herpes virus, you then manage to get under control, but what the virus actually does then is hides out in the central nervous system. And it basically hides there until it senses that the immune system is not as alert as it should be. And once it senses that the immune system is suppressed, it then reactivates and causes reinfection. And this is the sort of example where you have the primary infection at the lip, and then what happens is at the latent phase, the virus will be struggling to deal with the immune response, and what it does is it manages to then make its way up the ganglion or the nerve, and it'll stay in that nerve until that individual's immunosuppressed again. And one of the reasons that these viruses tend to hide in nerves is the fact that your central nervous system has a state of immunity called immune privilege. Immune privilege basically is a situation where the immune response in the central nervous system is at a very, very, very low level. There is still an immune system there, but it's very much a lot, at a much lower level compared to systemic tissues. And one of the reasons for this is the central nervous system wants to protect itself from an excessive immune response. And of course, some of these viruses manage to take advantage of that. And then you have, so we've talked about acute, we've talked about latency, and then we have persistent infections where some viruses just can persist and basically, tolerate the associated immune response, for instance, hepatitis B. And that whole concept leads to the idea of immune evasion by pathogens. And one concept is immune variation. So this is the basic structure of protein. You have the protein code, I suppose, and the ligands that are expressed on the outside, which help attachment to the cells. Then you have the nucleoprotein, the genomic RNA, and the matrix protein. So that's a sort of typical picture of a virus. Now, in terms of how some of these bacteria or viruses manage to evade the immune system, one concept is antigenic drift. Okay, so in the adaptive response on the left-hand side, you can see that the pathogen has a virome, and basically we produce antibodies that are able to easily identify those PAMPs, if you like, those pathogen-associated molecular patterns, and because we can recognize it, we can deal with the infection. And then we have the immune memory, which means we can keep it at bay. But what can happen is that some of these pathogens can mutate, and what that means is when they mutate their genetic material, then the components, the PAMPs that our B cells are used to recognizing change, which means the antibodies we previously had are no longer effective. That's called antigenic drift, and could be just through a simple point mutation. Whereas antigenic shift is where you have a situation where you have two pathogens, and one might have antimicrobial resistance, for instance, and they transfer genes to one of the pathogens. And what will happen in this situation is it's transferred genetic material which will enable the pathogen to change the expression of the PAMP. And same again, what will happen is because it's changed the PAMP, the nature or structure of the PAMP that's being expressed in the surface, our antibodies are no longer able to deal with to recognize the pathogen in question. And this has come with pneumococcal serotypes, very difficult. Now, what I'm going to cover now is immune evasion by pathogens in terms of inhibition of antigen presentation. Of course, everything to do with the immune system is very much relies on antigen presentation, because every adaptive immune response more or less requires a CD4 helper T cell response. And a lot of viruses in particular have adaptive ways to avoid this MHC presentation, particularly the MHC1. So some viruses encode proteins which inhibit the proteasome. So if you remember, the immunoproteasome is the organelle in the cell that's responsible for degrading the viral protein into short set sequences containing the epitope. So the fact that some viruses are able to stop that process means that the epitope will never reach the MHC, which means the epitope will never be presented on the surface of the cell. And if that happens, then the virus will get a free run in the cell, and you'll never have that process of antigen presentation. Other viruses, such as CMV and herpes, can inhibit the top receptor. And of course, the top receptor is pivotal to transporting the epitope of the virus into the endoplasmic reticulum and subsequent loading onto MHC1. So the fact that some viruses can inhibit that process will mean that that virus never gets presented. And there's other viruses as well that will inhibit MHC1 migration to the cell membrane. Of course, if you stop that migration to the cell membrane, then we have a problem because the MHC will never reach the surface, which means the T cells will never be able to recognize that there's a virus present. Okay, and then lastly, there is some viruses that manage to actually physically degrade the MHC1, and that will subsequently prevent presentation. And from I suppose Emer's lectures, you would have came across the concept that if a cell downregulates MHC1, then natural killer cells will kill that cell. Interestingly, what the CMV virus can do is produce its own version of MHC1, and it'll express the MHC1 on the surface so that the natural killer cell will not sense that there's a reduced not MHC1 expression, and therefore it will be able to survive. So that's kind of a neat way to avoid the innate response against viruses. Okay, so just to round things off then, just to introduce the concept of vaccination. You know, this is, I suppose, the exploitation of adaptive immunity. And it's the idea that we could give a suspension of attenuated live, so sort of weakened or killed microorganisms through an injection. So you're basically injecting a very weak pathogen or a dead pathogen through injection or through could also be drops to the human. And when you expose it to these weakened versions or dead versions of the microbes, then it basically makes it easy for the immune system to play about with it and get to grips with it and recognize it. Okay, and it can also be a modified bacterial toxin to enable the immune system to develop neutralizing antibodies against it. So some examples of live attenuated vaccines would be the MMR. And one caveat to all this is that when you're dealing with live attenuated or weakened vaccines, you need to be careful with people who have weak immune systems because it could have the potential to create problems. Okay, so you could also have recombinant vaccines or protein subunits where basically the components of the pathogen are isolated and injected as a vaccine. Okay, and the whole concept, of course, is that you give the vaccine, and because it's weakened or dead, the immune system can develop its primary response without having any threat of injury, which means then when the real McCoy comes along for the secondary response, our bodies are able to respond rapidly. Okay, so in terms of vaccination, you have active immunity, which results when lymphocytes respond to a vaccine or toxoid, and that results in the induction of memory. Okay, but another form can be where you get preformed antibodies. This is passive immunity, where you give preformed antibodies injected into the body, and the antibodies will have an effect. And actually, quite recently, this was one of the concepts involved in Donald Trump's treatment for coronavirus, where he was taking monoclonal antibodies targeted at coronavirus, and that won't give him long-term immunity, but it will certainly help his because he has the infection, it'll help reduce the viral load and make it easier for his immune system to deal with it. Good old Donald, eh? And then we have herd immunity. And herd immunity is a very important concept, and of course it's very topical at the minute. But it's basically a concept where enough people are immunized in the population to prevent the further spread of the disease to people who haven't been immunized. And generally it can be over 75% of the population are required to be immunized. But this can differ depending on the level of I suppose, spread or infectiousness that the particular pathogen can have. And this is a nice diagram, I suppose, to highlight the concept of herd immunity. So at the top section, you can see all the individuals in blue they haven't been immunized. But we have two people right in the middle there who are infected, and they're sick and contagious, which means all the people in contact with the red, the people in the red will then transfer to the surrounding people. So once the surrounding people become infected, they'll then spread it to the other non-immunized people. And here you can see that the disease will then subsequently spread very quickly through the population. Taking it down another level, if some of the population gets immunized, then the degree of spread isn't as severe, but it's still not effective enough. And that's why the level of spread is strongly influenced by the level of immunization. Because as you can see in the bottom situation, where the yellow folk who are immunized, when they're in contact with the red person, the infected person, then the red individuals who are infected can't spread it to the blue individuals, because the idea would be the red person should be able to spread the virus or the contagion to the yellow person, and the yellow spread it to the blue. But that can't happen, of course, because the yellow individuals are immunized, and if the pathogen does manage to enter the system of those immunized individuals, it'll be eliminated, and that will be the stop of the spread. And that's the concept of herd immunity that you can see here, where if someone gets infected, for instance, these red individuals, they actually can't spread it to people who are not immunized. And in terms of, okay, so what percent of population do we need to achieve this concept of herd immunity? It actually differs depending on how infectious the disease is. I'm sure you've heard of this, the R number, more recently. The R number is basically how many people an infected person can spread the virus to, spread the infectious agent to. So as you can see here, mumps, the R number is 4 to 7, polio 5 to 7, smallpox 5 to 7, diphtheria 6 to 7, rubella 6 to 7. And the percentage required for herd immunity threshold sort of ranges between 75 and sort of 86 for that sort of region. But as you can see, when you get the other diseases such as pertussis and measles, where the infectious rate is a lot higher, because the infection rate's a lot higher, then the threshold for herd immunity is a lot higher then as a result. I think for coronavirus, there's a lot of discussion about, what's the R number for a start. It's variable depending on, the way in which the public interact with each other. But there's sort of initial indications that the R number for coronavirus is somewhere in the region between 2.5 and probably 4. But I think it's around 2.5. And the suggested threshold has been as low as sort of 60%, and even lower in some estimates. So sort of between, I think it was 45 and 60% of the population would need to be vaccinated or have immunity to enable us to achieve herd immunity. And that's probably because the coronavirus isn't as infectious as some of these other components. Okay, folks, so that's the first presentation done. And then, as I say, the next presentation will be a lot more technically challenging because it goes into a lot more detail. So thank you and good luck with the next presentation.